Now it's my great pleasure to introduce Mark Lintern. Mark is the author of the book, The Cancer Resolution. And The Cancer Resolution is the result of his 8 years researching and burrowing into what is going on underneath the hallmarks of cancer. Our conference is all about metabolic flexibility. It's all about how cancer cells manipulate their environment, how they thrive, how they find their way around all the orthodox treatments that are thrown at them. And Mark's book is bang on time and trend in what we need to move our thinking together forward. I think this piece of work, my personal view, is that it's going to be landmark in how we understand and deal with cancer, up there with Jay McClellan's work, up there with Siegfried's metabolic theory. It absolutely is enabling our understanding. I'm using it in practice, and we are, seeing a resolution coming through in many cases that we haven't before. So I'm super excited to be speaking with Mark today and have him explain everything that's the result of his extremely dedicated and hard work. So thank you very much for joining us, Mark.
Thank you, Patricia. It's absolutely pleasure to be here. Thank you.
And we're so looking forward to seeing you at the conference and getting your input, and hopefully you signing your book and everybody really getting to know about your work, because it's of vital importance. So tell us how you, a graphic designer, if I may say so by trade, came into, dedicating 8 years of your life to this phenomenal piece of work.
Yeah, it was unfortunate, really. And I think a lot of people who come into this field from outside of it, it's because of unfortunate circumstances. So for me, it was a diagnosis myself. And initially, I asked my oncologist and doctor what were the DNA genes that caused it, because I wanted to prevent it in future. I was lucky; I had it surgically removed. It was a lump, a skin cancer, and I caught it early, so that was fine. But I still wanted to prevent it, so I wanted to know which DNA genes caused it and what external influences could have led to it, those genes being damaged, and they couldn't answer those questions. So I started my own research, trying to delve deeper into the science behind it, to try and understand it. But the more I got involved, the more I realized how complex it was.
Mm-hmm.
Also that scientists were still themselves struggling to understand this complexity and struggling to identify any particular patterns that could explain why. So that confused me somewhat. And from that initial research, I got to hear about several other theories, including the metabolic theory. So I realized quite early on that cancer wasn't necessarily, or scientists don't all agree on what the cause of cancer is. It's not necessarily caused by genetic mutations. There's confusion. There was mass confusion in the industry. And that surprised me somewhat, because from my oncologist and my doctor, and from what I've known from hearsay, hearing about cancer before I was diagnosed myself, was that it was definitely a genetic disease. The impression I was given from what I'd heard was it was definitely a genetic disease. So this initial research that I did lasted about 4 or 5 months, and that really opened my eyes to the fact that actually we don't quite understand what the disease is. And then unfortunately, a couple of years later, a friend of mine got cervical cancer at the age of 30, and I then decided, you know what, I'll go back into the research that I did and try and help her as best I could. But with her, it was difficult, because I was fighting this notion that we should just listen to the oncologist and follow what they do and say. So my friend was between a rock and a hard place, really, listening to some of the research that I was trying to present her with looking at the disease in alternative ways, looking at the disease through the metabolic theory. But she was also torn between, trusting the fear that she got from a diagnosis as well, of course, and then feeling like she could only really trust the doctors to do right by her. So it was a struggle to get this information to her and show that there was a different way of potentially approaching the disease alongside conventional treatments. Unfortunately, she passed away after a year, and during that whole time, I did a whole year's worth of intense research, and it just exploded my understanding of the disease from many different perspectives and the limitations of conventional medicine. And from that, because Sam unfortunately passed away, and there was a lot I could see that was wrong with the way she was treated. Not necessarily, she wasn't treated nastily. The nurses and the oncologists were lovely, fantastic people. They're doing their best in difficult circumstances. But it's just some of the evidence that I found and some of the treatments she was being offered, it was clear to me that they weren't going to work. So this made me very angry. And I thought, well, I'm not going to let Sam die in vain. I'm going to continue my research, and my current findings I'm going to place within a book. The initial idea was for me to present my findings in a way that my family and friends could understand it. And it was just to be a little booklet that was to show anyone who I cared about who developed it, they could literally read it and realize that there is an alternative view, and we could target the metabolism of cells. So it was to help them, like I would have liked to have helped Sam in that process. So I started that, and as I delved deeper into the research, I went down a rabbit hole. Started realizing there was a little bit of contention with metabolic theory. I then started looking at other theories such as the atavistic theory, tissue organization field theory, bacterial theory, speciation theory, aneuploid theory. All these theories, I just thought, I'm really interested in this now. It kind of took over my life a little bit. It just went on from there. And 8 years later, I came across several aspects from my research which pointed me in a particular direction and enabled me to highlight an alternative causal factor in the Warburg effect, which I think is absolutely pivotal.
Yes.
The Warburg effect is pivotal in the propagation of cancer. Mostly the hallmarks appear to be a downstream event of the Warburg effect. So hallmark 7, which is abnormal metabolism, seems to be the key factor to explain. And I came to that conclusion after several years of doing the research, that this was a key aspect of cancer that would need to be explained. And if you could identify the underlying cause of hallmark 7 or the Warburg effect, then this may take us on a different direction to possibly identifying another cause, if not the cause of cancer itself.
That's absolutely right. I was just reflecting as you were talking there on my own journey, as it were. Sort of 35 years of working with cancer from being in the orthodox side, and I've seen those. I came from a point of view of not understanding much and not being able to answer people's questions and being curious. And I've seen, the use of classical chemotherapy, which always promoted metabolic flexibility and diminishing returns and side effects that weren't adequately dealt with. I remember when they started mapping the genome and the massive interest in, we will know the genome pattern for each type of cancer, and then the kind of horror when they realized that 2 people with pancreatic cancer do not have anything like the same genome patterns whatsoever. So gene therapy raised its head and then slowly went away. And
then you have the heterogeneity within the same tumor.
Absolutely. So individual. As you peel the layers of the onion, you find that there's so much going on there. And then this kind of singular pursuit of cancer treatments from the orthodox side, ignoring the metabolics, ignoring the microbiome, ignoring the extracellular environment in which cancer cells in. Ultimately, as we've just seen repeated, Mark, haven't we, that it ultimately ends up in the same place, that you get some benefits, certainly, but ultimately runs out of steam. Metabolic flexibility. And what's wonderful with your work, and I'm seeing emerging now, is you ignore it at your peril. Immunotherapies won't work very well if the gut's not in a good place, if the gut bacteria is not good, if the immune system's suppressed. It sounds ridiculously simplistic, but that's it. And everybody who's involved with the conference, this is absolutely all come back to the same element, that you need to work on the environment of the body, on the cellular environment. You need to take care of these other things if the treatments are going to work, which is absolutely right. And so tell me more about the viral bacterial elements to this, that's the fungal side of your work.
Yeah, so essentially, as I was going through, I'd identified, I'd contacted Thomas Seyfried early on in my research, and he gave me some pointers on what I need to be looking for. So he was the one who introduced me to the hallmarks of cancer. And because he said he was in the process of attempting to explain the hallmarks with the metabolic theory, and that the metabolic theory can explain all 10 through the mechanism of defective OXPHOS. Now, so I took that on board. I studied the hallmarks, and I realized that if I wanted to create any sort of credible theory, I'd have to find the evidence that could explain all these different hallmarks. But by this point, I hadn't identified any particular pathogens. I was still looking at different theories. At first, I thought cancer was caused by viruses. So I'd gone down the viral theory route, but then there were contentions there. The same with bacteria, there were contentions there. And then I stumbled across an immunological paper, because I was just interested in generally how the body works from all different perspectives. So I was interested in how infection affects the body. And in this one particular paper, I stumbled across a mention of the Warburg effect. So it highlighted that when particular pathogens, including bacterial and fungal pathogens, as well as viruses, when they turn opportunistic and try and invade the cell actually responds with a Warburg-like response. Now, that's literally the phrase that is stated within this paper. And I stepped back from that, and it was just like a light bulb moment for me, because a lot of the conventional thinking is that the cell is actually damaged and it malfunctions.
Yes.
And that damage then results in autonomy. The cell develops a mind of its own, and it's like turned into this evil entity that's trying to just propagate by itself and grow. So it has a mind of its own. However, this paper was suggesting that, hold on a minute, here's a key factor of cancer, which is essentially the Warburg effect, the switch in energy to glycolysis. And it's instigated by pathogens. And at this point, I was also reading up a lot on the microbiome, as you just mentioned, and how important that is to our health. And from my research, to me, it's critically important to our health.
Indeed.
And that's one of the things that orthodox medicine doesn't really take into account. I mean, chemotherapy destroys the microbiome. So with that in mind, I realized that, okay, well, how prevalent are these microorganisms within the human body? And the more research I did, it became clear that the body isn't as sterile as we originally thought. These microbes can migrate into the pancreas, they can migrate into many different areas of tissue. And so that took me on the trajectory of thinking, well, the hallmark 7 and cancer metabolism, Warburg effect, is key. And there's contention over that. Professor Seyfried will claim, that defective OXPHOS is the underlying mechanism that drives this Warburg effect. But I've noticed that others contend this. So I'm in a position where I'm on the fence. I'm trying to be objective, and I'm seeing this contention. There's no point in me siding with any particular one.
Yes.
I may as well take this evidence forward. And I thought, well, being as though this has been mentioned as a Warburg-like response, surely this has already been assessed by scientists, which is why no one's really picked it up. So I didn't think much of it at that point. But then I was trying to do research into the Warburg effect being created by infection within cancer circles, within oncology, and there was no mention of it. There was absolutely no mention of it. So I began to realize that people weren't looking at this aspect because they were so focused on the cell malfunction side of things. So that then drove me to try and understand how infection can manipulate the Warburg effect and whether or not other aspects, other hallmarks can be explained by the infectious process. And then that led to more information in me discovering Ravid Straussman's work, where he highlighted that now recently all tumors that he's investigated, along with his colleagues as well, that tumors harbor their own tumor-associated microbiome, so pathogens, particular pathogens that are present within those tumors. So that corroborates the idea that pathogens are present. Now, they could be present after the cause, after the fact that the tumor has developed. They could just colonize the tumor, so they could have no impact. But the other aspect of that is, well, actually, they could be an underlying factor that's driving the disease. So there's two aspects to this. So we can't just dismiss one side because we think that they might have colonized the tumor afterwards. And then the more I looked into this, the more I realized there's a lot of research out there that shows that particular pathogens are actually driving the disease, particularly in pancreatic cancer. Yeah. So there's a lot of evidence for that. So then that made sense to me. So now I realized that I've got an explanation for, probably an alternative explanation for the Warburg effect, one of the most key aspects of the disease. And then as time went on, I was able to explain all the other hallmarks and many other conditions associated with cancer besides those hallmarks through this control mechanism, this suppression, if you will, of the cell by intracellular pathogens. So that's where it took me. And this whole process of looking at the disease through this cell suppression lens, where the cell itself isn't at fault, it's not developed autonomy, it's trying to do the very thing that it should be doing, is trying to protect the host. However, what seems to have happened is that a parasitic intracellular pathogen has gained control of particular pathways, incidentally, the same pathways that we see manipulated metabolically in cancer. Yeah, absolutely. And this can explain why we see these metabolic pathways change in the way that they do. I obviously didn't find this out straight away. It was a slow process of realizing that the Warburg effect is key, that many aspects of the hallmarks are downstream events of the Warburg effect. But then this whole idea that intracellular pathogens can trigger the Warburg effect was fundamental. Mm-hmm. The very interesting thing about it is that I noticed Thomas Seyfried states that he admits that cancer cells are absorbing oxygen at quite high rates.
Yes.
But he argues that they're not using it through the OXPHOS pathway to generate ATP. He highlights that they're creating excessive amounts of ROS, reactive oxygen species. Now, the interesting thing is, as I was doing my research into infection, I came across Dr. Robert Navio's work, where he posits his cell danger response model, which discusses exactly what happens when a fungal pathogen or any particular type of pathogen enters into a cell. And what occurs is that the cell chooses to switch to glycolysis, chooses to mm-hmm. switch to the Warburg effect, yeah, in order that it can utilize the oxygen to flood the cytosol with oxygen in order to inhibit the pathogen's ability to produce RNA, proteins, and DNA. So it's an inhibitory response. It's an anti-infection response. So the cell is now utilizing the oxygen, intentionally switching OXPHOS off in favor of using the oxygen to combat the pathogen. That, of course, leads to the glycolysis whilst the pathogen is present. Now, if the cell fails to eliminate the pathogen for whatever reason, the immune system is weak, then the Warburg effect will remain until the pathogen is removed. Now, that for me helps to explain why the Warburg effect occurs and why it's a prominent feature of the disease. And then when you take Ravid Straussman's work, where he highlights that intracellular pathogens are present within all tumors, you can kind of understand why the Warburg effect persists. Not only that, but Ravage-Straussman highlights through a process of purinogenic signaling that you don't have to have all the cells within the tumor infected. What you have is a feedback from the cells that are infected to non-infected cells in the periphery. They all triggers the Warburg effect, even though the cell isn't infected. In other words, the cells are working communally to initiate the antimicrobial infection strategy, which is the Warburg effect. Now, that will remain until the pathogen is eliminated. Now, for various reasons we can go into, a tumor environment seems to benefit the pathogen and seems to suppress the immune system. So within that little tumor niche, you have this problem where the immune cells are blocked, essentially, or suppressed from actually accessing those cells.
Absolutely. Yeah.
Because of the iron overload, because of the excess lactic acid that's being produced. And then from all this research, I began to really formulate the consistency, explaining the consistency of the disease. Because one of the big issues with me looking at the disease initially was that cancer, through the hallmarks, is a consistent disease. These are consistent attributes of fundamental attributes of the disease. But what we're told through the mechanism of cell suppression that all the other mainstream theories abide by is that any particular random DNA damage, any particular damage to the cell, to the respiration, can cause the disease. But the problem with that is that randomness cannot, in my book, cause consistency.
Indeed. Yes.
Yeah, one of the main arguments and the main problems I had is that we're trying to say that any random combination of damage can cause the disease. However, the problem with that as well is that we haven't been able to highlight or replicate any particular damage, random damage, to recreate cancer in that way. That was a big red flag for me. So this notion that we can generate the disease through randomness suggested to me that actually there's a consistent factor involved, which I'm now pointing out is the fungal pathogen that is present consistently within the cell and triggers the Warburg effect. That consistency leads to the consistent conditions of cancer, and that the randomness that we see, the random DNA damage and the random metabolic output, is a symptom of the toxins and the battle that we have between the stubborn parasitic pathogen that's present within the tumor mass. So yeah, there's definitely so it's worth looking at this other perspective is all I'm really saying. I'm not saying that I'm particularly right. It's just that we need to be, there is another a different explanation for the Warburg effect and an actual reason why the cells are acting the way that they are. And this is the cell suppression idea that I'm pushing, rather than the cell malfunction idea that the cell's gone wrong and rogue. Because there's one other aspect that kind of highlights this to me is that when I was looking at the research, I found the evidence where natural substances were able to initiate selectively apoptosis in cancer cells as opposed to healthy cells. And I'm always trying to ask particular questions that I noticed that sometimes aren't being explained. So my question would be, why? Why is that occurring? We know that certain natural products, compounds can benefit cancer. We know this, but we don't know why. So I've been trying to ask why. It's selective, and the answer I would have for that is that particular organic compounds are antimicrobial, and what you're seeing is a response to these compounds where the intracellular pathogen that's affecting the tumor cells is killed, and the suppression it has over the cell is relinquished, and particular pathways resort back to normal, or apoptosis occurs if the cell is damaged beyond a certain threshold. Now, these compounds, natural compounds, act selectively. They do not act upon the same pathways in the same way in healthy cells, because there is no parasitic pathogen affecting those cells in any abnormal way. And that would be how I'd explain it. There's one particular study that shows that within cancer, MMP9 is upregulated, and they're not sure why that is, but they're trying to target it. But then there's this other study where you're looking into infection, and what you find is that particular pathogens, fungal pathogens in particular, Candida albicans, influences the inhibitor of MMP9, TIMP1, by suppressing it, which allows the pathogen itself to stimulate MMP9 production. So there's that aspect. So you've got, okay, hold on. So in cancer cell, you've got MMP9 production is overproduced. You've got the fungal pathogen, which can overproduce that. But then another study shows that if you use cannabis oil on cancer cells, what you find is that TIMP1 is reduced, increased, and MMP9 is reduced. And so the conclusion drawn is that cannabis specifically targets that pathway directly, and it could do. But what I'm offering is another explanation to suggest that doesn't occur in healthy cells, and it doesn't occur in healthy cells, and it only occurs in the cancer cell, because what's happening is the cannabis compounds are killing the fungal pathogen that is actually controlling and upregulating MMP9 and suppressing TIMP1. So when you kill the pathogen with cannabis oil or whatever compound it is, that then kills the pathogen, then the cell reverts back to a homeostasis and TIMP1 is upregulated. So that would be my argument for that particular reason why we have that change when using those compounds. I'm obviously not saying that compounds do not directly affect the cell, they do, but there is this alternative potential explanation that needs to be investigated.
Yeah, no, for me, it absolutely makes sense, because whilst a lot of integrative medicine has approaches have kind of been developed in perhaps a trial and error way, or through certain methods, a large part of it has been, as you say, antifungal, antibacterial, not necessarily by design, but as a product of things that you're trying to do elsewhere, with trying to upregulate the environment and upregulate genes, etc. So to some extent, this really highlights that, we know that by working in this way, you can make a difference. We've not all, perhaps I've not been scientific enough to understand the specifics of that. And I think this adds a very large part to the explanations of that. And also, the constant cycle of people being treated for cancer with standard of care treatments where you just see diminishing returns. And that feedback loop, that cycle doesn't stop, that as soon as the treatment stops, it's okay, let's stand back, wait, see what happens, and what happens is recurrence. And that is absolutely an area that needs to be addressed and taken seriously. So, my great hope is that the orthodox world will look at everything that's going on in the integrative world and say, actually, yes, there is absolutely a fit here, that we need to do these processes alongside standard of care to, so that they can be used in an effective way that doesn't actually promote metabolic flexibility, which unfortunately at the moment it does. And for me personally, it makes no sense whatsoever to use standard of care exclusively. Yeah,
no,
absolutely. I think it's just setting up the potential, isn't it?
It just makes sense to look at the microbiome, because that's just fundamental.
Yeah.
And like what Dr. Nacia Winters is looking at with the terrain of the cell, you've got to approach the metabolism of cancer. You've got to approach the terrain. You've got to approach the microbiome. And when you do approach all those things and bring homeostasis back to the terrain, you're looking after the cells, removing all the toxins and allowing the cells to reduce inflammation. And when you're targeting the microbiome as well as the metabolism side of things, what you're actually doing as well is you're making it more difficult for fungal pathogens to exist within that environment. So on top of those three modalities, targeting metabolism, targeting the terrain, and targeting microbiome, I'm suggesting that we also try and mindfully target additionally, in as less as a toxic manner as possible, an antifungal approach.
Yeah, totally.
A lot of the drugs that I see that the Care Oncology Clinic uses, and a lot of the drugs I've noticed when I've done my research that seem to be effective, off-label drugs that have been used by Jane McClellan, for instance, lovastatin, even tamoxifen. These drugs are antifungal in their own right. Even doxycycline is antifungal. So there is this underlying aspect to all these potentially effective mechanisms we're using, which is that they are all antifungal as well. So you've got to ask if that's a major aspect to it. Are these antifungal aspects and antifungal drugs, which seem to be effective against a broad range of cancers, are they just acting upon particular pathways that the cancer cell is using? Is the cancer cell got a mind of its own, it's using these pathways and they're being affected by the antifungal drugs? Or is the cell trying to do its best? Is it being suppressed by a particular fungal pathogen? And do those antifungal aspects of these drugs attack the pathogen directly, which releases its suppression over the cell, resulting in apoptosis or remission?
Absolutely. And I think that research has to, as you quite rightly point out in the book, this has been there in research for a while, but nobody was looking because of the way that we look at cancer traditionally from the orthodox perspective, the way that we view it, and the way
very segregated, isn't it?
Very, very one-dimensional, really. And
but I understand that because it's such a complex, DNA is such a complex area to study, and metabolism is such a complex area. So I understand why it's gone down that route. But in order to solve such a complex problem, there needs to be crosstalk between all different disciplines.
Absolutely. Another of our great contributors to the conference is Professor Serge Joaginalis, get that right, who very much focuses on how P53 suppression and P53 mutation contributes to cell mortality. And it's very much, the two things that are common with both, your theories and his theory, is that there is nothing about standard of care that is going to tackle metabolic flexibility, that is going to tackle how cells just continually come back and survive and then thrive. And it's absolutely spot on that if we don't work on this and change the environment, then people are just being set up for treatment failure, unfortunately. So I think, as I say, I think your work is seminal. None of us are claiming that any of our work is kind of perfect, but my goodness, there's a very strong element coming through now, isn't there? Functional testing is allowing us to look very, very closely at what's actually happening with cancer.
Yeah, absolutely. And I think even Robert Weinberg, the co-authors of the Hallmarks of Cancer, he's highlighted that if you just use standard of care, you're not going to knock out the cancer stem cells. So we need to be approaching things from a different perspective. I mean, Professor Max Witcher as well, he's another key cancer stem cell proponent, and highlights the same. And even he stipulates natural compounds in, say, broccoli, sulforaphane and what have you. He highlights how they affect cancer stem cells, and that you have to use, if you're going to use conventional treatment, you have to use other treatments as well, other modalities that can target different aspects of metabolism and cancer stem cells as well, because you're just going to reduce most of the tumor, the regular cancer cells, but you're going to stimulate the cancer stem cells. Yeah,
you're going to create an environment that cancer cells actually thrive in, which is and you're not going to repair the genes like the PTEN gene, which switches metabolism over very, very much to choline. All this evidence is there of the sequence that's happening, and not enough is being done to stop that sequence by any stretch of the imagination. So
I mean, you mentioned there that you're creating an environment that cancer cells can propagate in, but that environment is also an environment that benefits fungal pathogens. They propagate in that. So it's
exactly that.
The similarities are quite incredible.
Yeah, no, they absolutely are. And as I say, I think it's I so admire you for doing this. I think it's a seminal piece of work, and I think in time it will be absolutely seen that like Jane's great work, that's a big step forward to our really getting to understand exactly what people need to help to deal with cancer, which is more wider approaches, more focused on exactly what the feedback loop is.
Yeah, and I think just to have a conversation about a different view, and just to get everyone to step back and question a current approach, because it's clearly it's beneficial for some, but it's not beneficial for most. So it's not working. We're struggling to make progress as we have done against other diseases. So we do need to step back. If only just gets us to reevaluate and look at things from a different perspective.
Absolutely, that. Get everybody around the table, take a big step back, and almost sort of say, right, okay, let's just go at this from a completely different route. And I think it would be amazing.
Yeah.
The results of that.
The thing is, I'm not saying I'm right. What I'm saying is that there's this different way of looking at the disease, and please challenge what I'm saying. You know,
we're all open to that. Absolutely,
science,
aren't we? Yeah.
Challenge it. I want people to take my theory through the mill, point out where it's areas that are in contention, and really go at it, because this is the only way we get to the truth.
Yeah, don't discount these theories, and scientifically, you know, we're all looking, we're all curious, and scientifically everything should be challenged, but at the moment, you know, mainstream is not being challenged sufficiently to open their minds to the whole metabolic process, and they really need to be. Absolutely. Mark, thank you so much. Thank you so much. Amazing. I know you could talk about this for probably about 5 more hours and cover for more content. But
days and days. It's all I've been doing for 8 years. I need an outlet. So it's been an absolute pleasure talking to you, Patricia. It's
been fantastic. You know, everybody should get a copy of your book, The Cancer Resolution. It's a seminal piece of work. And you know, I often say to my clients when I'm working with them is that you will understand more about cancer, and you will understand more about yourself and what's happening than your oncologist will. And, you know, I think this is a major contributor to this. So I can't wait to see you in September. You were there signing your book. Fantastic. And, you know, exciting days ahead. So thanks so much, Mark. It's been brilliant.
Thank you. Take care, Patricia.
Thank you.